All right, so welcome back. We're on to vision. All right, so I think we saw this in the homunculus, but right, a good chunk of our cerebral cortex is actually devoted to processing visual information, right? And we talked about that where we have a separate brain region that's just responsible for recognizing people's faces, right? Vision is going to be very important to us. So the part of the electromagnetic radiation that we can see is very small. It's limited to wavelengths of 400 nanometers to 700 nanometers. And that's what we call our visible light, right? That's the Roy G. Biv that we're all familiar with. When we talk about wavelength, right, light is going to be a series of waves, right? And that's going to be that distance of the waves that we're going to be able to pick up on. Those the receptors in our eyes are sensitive to. Now, just as a little bit of a aside for you, different animals can actually pick up on UV and infrared rays. So for example, snakes and their pit organs, like the viper, you'll notice them sort of like here on their mouths, they're able to pick up on infrared. So it's usually what we think of like heat information. And actually those neurons will actually go into their occipital lobe as well, where all the visual information goes. So it's thought that, they can see just fine, but then they have this overlay of like temperature on whatever it is that they see. So maybe they see like the grass, but oh, there's a warm spot right behind it. That's probably a mouse behind it. Or if it's not, if it's cool, right, it's a rock, right, that kind of thing. And then a lot of birds actually can see into the UV. So you use a black light, right, and something glows, that's UV. So birds, when they look at, let's say they look at, all right, let me back up. We look at a ground outside, right? Grass, rocks, dirt, whatever. That's what it looks like to us. Certain birds of prey, hawks, owls, look at that and they see a bright streak where a mouse had peed. Because that happens to fluoresce. I don't know how exciting that is to blacklight, but they can see it. It actually glows for them. Certain things like bees, there's actually UV designs on a lot of flowers. So like, and I can't think of an exact type of flower, but so let's say these are the petals of the flower. There's actually a design in each one of the petals. So bees can actually see that individual design. I had this advisor when I was an undergrad who joked that like penguins, how like emperor penguins, they all look alike, right? But they have those big white chests. What if on those big white chests they actually had something in the UV range that when the females come back or the males come back, they actually look at one another like, oh yeah, I could totally tell that was Bob. It says so right there. Bob. Right? We just can't tell them apart because we don't see into the UV. As far as I know, they haven't done those kinds of tests yet, but if they should, it'd be so interesting. Anyway. Alright. I will hide me so you can see these images better. Alright, so let's just talk about some of the structures of the eye then. So eyelids, another name for eyelids are the palpebrae, right? That's plural. Palpebra is singular, right? It's there. I think we all know it helps protect our eyes. That's obvious, right? As we blink, we're going to move tears across our eyes, so it helps lubricate our eyes. I think we know that too, right? If you keep your eye open too long, it dries out, right? Pretty obvious. Okay. We have what are known as tarsal glands. So these are modified, or these might on here, well, I'm sorry, they're called meibomian glands on this image, so in green here. These are modified sebaceous glands. They basically prevent your eyelids from shutting together. That's what their job is. Okay, and then the conjunctiva, and you have what's known as the palpebral and bulbar versions of it. So I don't know, if you kind of look at your slide, right on the underside of your eyelid is sort of this orangey thing. And then you can kind of see it loops around here and it goes on the eyeball itself. So there's a palpebral conjunctiva and then the bulbar one. So basically what this is it's a transparent mucous membrane. And basically what it's doing is it's helping to produce lubricating mucus that helps the eye from drying out. It also is where your blood vessels are. So blood vessels actually are found in the conjunctiva. So when you have bloodshot eyes, it's actually the blood vessels in this membrane that are being bloodshot, not in the sclera. Okay. All right, I think we all know what your eyebrows and eyelashes are. You'll have some sebaceous glands. They're found at the base of your eyelashes. Mentioned those a little while ago as well. So when those get impacted, there's some sort of bacterial infection. That's when you get a sty. That's what that is. Okay. And we've seen this term in lab. Maybe we have by now, maybe we haven't. But a palpable fissure is simply the gap between your eyelids. So you can kind of see here, that's gap between the eyelids is simply the palpable fissure. So obviously if you're squinting, that fissure is very narrow. If your eyes are wide open, the fissure is much bigger. Okay, so it'll vary depending on how open or closed your eyelids are. Okay. We have something known as the lacrimal apparatus. So the lacrimal gland is going to be located on the superior lateral portion of the eye, so just underneath that superior orbital margin of the frontal bone. Okay, and it's what's producing tears. So you can see this, produces about 1 milliliter of tears a day, right? And then as you blink, you spread it over your eye. And interestingly enough, it has an enzyme that will basically kill off any kind of bacteria. Now, obviously, this doesn't always work. We do get pink eye, but for the most part, our eyes, tend to be fairly infection-free. Okay? So basically, this will produce the tears. They get spread over your eye, and then right here in this little pink thing in the corner of your eye, what's going to end up happening is the tears flow over your eye, and then you have 2 little holes, and then they're going to drain into the inferior and superior lacrimal canals, into this larger structure called the lacrimal, nasolacrimal duct, and then basically into your nose. So I think I mentioned this to you before. What ends up happening when you cry is that you produce more tears, and they go down that, remember that lacrimal canal? We saw that in the skull. So basically that's where these tubes are. They go down that lacrimal canal into this duct, into your nose. So again, that's what allows you to have that awesome sniffing thing. All right. Oop, lost my cursor. I wanted to mention something else to you. Oh, just want to mention one last thing to you. That actual pink thing here is going to be called the lacrimal caruncle. You know how you get the white goop, right, that builds up right there? It's basically because it's producing a whitish, oily secretion. It's made up of basically sebaceous and sweat glands, and again, produce that white goop that after a while it kind of just crusts up on you. Anyway, that's what that's called, lacrimal caruncle. All right, so the eye's going to have different layers. Your different textbooks will give you slightly different things, but they call them the fibrous tunic, the vascular tunic, and then the nervous tunic. So the fibrous layer, vascular layer, and then the nervous layer. Okay. So we're going to take a look at those. So the fibrous layer is basically going to be the outer part, the white part of your eye, which is the sclera. Remember, it's made up of dense, irregular connective tissue. So collagen fibers, as well as those fibroblasts that are making them. And then the cornea, which is going to be the clear part of your eye. Okay, so this clear part of your eye. Right. You have an opening in between the two, like right here, right where my arrow is. There'll be a little opening. There'll be a drainage port called the scleral venous sinus, or as I like to better call it, the canal of Schlemm. It's the old name for it. My most favorite words in biology. Basically, we'll talk a little bit more about this. You're going to have a fluid made in this anterior portion of the eye that gets drained by the scleral venous sinus. Okay. The back part of the sclera is basically interrupted by the optic nerve. So you're going to have all these neurons inside here, and they're going to send their axons out the back of the eye. Okay. Pretty obvious. All right, so in the vascular tunic, you're going to have the iris and the pupil. The iris is going to be the colored portion of your eye. Okay, it's basically a flat donut between the cornea and the lens. So again, here's the cornea right here. This is the lens. The iris and the pupil are in between the two. So the iris, as I mentioned, is the colored portion of the eye. So if you have green eyes, brown eyes, blue eyes, violet eyes, hazel eyes, I don't know, whatever color, that's going to be the color of the iris. Okay. Again, it's donut shaped. So just like a donut has a hole in the middle, your iris has a hole in the middle, and that hole is the pupil. Okay. And the job of the iris is going to be either to increase or decrease the size of that pupil, and thereby regulating the amount of light that's coming into your eye. We'll see there's this other thing called the choroid that is a dark staining, that's a darkly staining part of the eye. Its job is to absorb any light that's coming into your eye. So the fact that when you look into someone's eyes and they have black pupils is because all the light is being absorbed by that choroid. What happens when you take a picture, for example, and you get red eye, the flash is so bright that the choroid can't absorb all that, and so what's happening is light is bouncing back out of your eye off the blood vessels, and so it appears red because of the red blood vessels in your eye. And we'll talk more about the ciliary body in a few slides. Basically what it's going to do is it will change the shape of the lens, helping you focus light onto the back of your eye. So if you look over here, light's coming into the eye, and you're going to try to focus it on the same spot on the back part of the retina, right back in here. Okay. So this is just what I mentioned to you before. So here's the iris, and I don't know why they made it this peach color. It's like an albino eye. They could at least give it some color so it looks more realistic. But you have two different sort of configurations of fibers. So some of these are going to be running in a circle, and then others are kind of radial. And basically you can kind of see here, so the pupil is going to constrict when the parasympathetic nervous system is active. And remember, parasympathetic nervous system is your rest and digest nervous system. So this is you right now watching this video. You're not, no zombies are attacking, so you're pretty much relaxed. Okay, so you don't necessarily need to have a ton of light going into your eye, right? However, let's say something happens, zombies suddenly invade, your eyes will dilate. Okay, that's to allow more light into your eye, to make sure that if you're running away or fighting off whatever, the zombie, you actually can see what's going on. Right? When you're trying to make your big getaway, is not the time to not notice that rock, like in other horror movies, trip over it and fall down. Right? So your eyes are going to dilate. Interestingly enough, when you're with someone that you actually really like them, your eyes should dilate. Well, and so should theirs. Their eyes should actually dilate. So if you're ever on a date and your date's pupils are like little pinholes, they don't like you. Don't stop dating that one. The other time that you'll have this reflex of sympathetic versus parasympathetic on your eye is if you shine light in it, right? So this is an autonomic reflex. So if I were to shine light into your eye, basically what's going to happen is your iris will constrict the pupil to make it little, so to protect your eye, right? Because there's tons of light coming in, it'll constrict. Versus under dim light, the eye will actually dilate, so the sympathetic nervous system will dilate the pupil so that it actually is bigger, so you can actually see better. So more light goes into your eye, you can see better under low light conditions. All right, also in this vascular tunic is the lens. It is avascular, meaning there's no blood vessels going to it. It has many layers to it, much like an onion would. So if you were to cut one open, you'd see that there's tons of different layers on it. In lab, if you did the sheep eye, that lens was very cloudy. That's due to the preservative. In real life, it's very clear, perfectly transparent. It's held in place by suspensory ligaments. And again, it's going to help focus light on something called the fovea centralis in the back of the eye. We'll talk more about why that is shortly. Another thing that happens with age is that because this is like an onion, it adds layers to it continually through life. So at some point, it has added so many layers that it makes it very difficult for it to focus. It becomes too burdensome, and it won't focus light on the back of the eye as well as it once did. So So again, with aging, the lens loses its elasticity and its ability to do something called accommodate. So what it's doing is that it'll change its shape from sort of like a fatter disc to a more narrow disc, depending on whether or not you're looking at something up close or far away. We'll see pictures of this shortly. So basically, when it's no longer able to do that, that's when you have presbyopia. Okay, so the inability of your eyes to adjust to reading up close, for example, anymore. It's an example of presbyopia. So these suspensory ligaments are going to pull on that lens. They're attached to something called the ciliary body, which is basically a muscle. And they're going to pull on that lens, and that's going to basically cause it either to be kind of stretched out or, a little bit rounder in the middle. Again, that will determine how things are being focused in the back of your eye. Again, we'll see some images in a little bit. It'll make more sense then. Okay. Your eye has different cavities to it. So there's an anterior cavity and then a posterior cavity. This anterior cavity is filled with something called aqueous humor. That aqueous humor, it's very fluid-like fluid. It's a terrible description. It's a fluid. Basically, it's produced by the ciliary body, and it's always being drained. So you get this kind of replacement every 90 minutes or so. Okay. In the posterior cavity, it also has a fluid in it, but it's much more jelly-like. Okay. You probably saw this also in lab when you did the sheep eye. It's only formed once. So when you end up seeing floaters, like, I don't know if you ever look at a particularly blue sky is predominant, you'll end up seeing these little floaters. It's debris casting a shadow on the back of your eye, and that's actually what you're seeing because it doesn't get replaced. So if there's debris in there, it stays in there. Glaucoma, what happens with this is that as you get older, right, you're still producing the aqueous humor just fine. However, that canal of Schlemm or that scleral venous sinus no longer drains the humor like it once did. So if you're producing too much of it, or I shouldn't say too much, but you're producing it at the normal rate, but you're not draining it like you once did, you get a buildup of fluid, right? You get a buildup of fluid, you're going to increase that pressure. You increase that pressure, you may end up causing damage to the retina, which is where all of our neurons are going to be. And if you're going to damage the neurons, that can lead to blindness. Okay? And the way that grandma's herbal glaucoma medication works, that's legal in Colorado, right, is basically what it does is it slows down the formation of aqueous humor. So by slowing down the formation of aqueous humor, there's more time to drain it, thereby relieving the pressure and thereby relieving the glaucoma symptoms. So that's how that works. Right. And then we have the last part is a nervous tunic. So that's the retina. Basically, this is where all of our neurons are going to be, right? So these are where our optic receptors are going to be. You have a region in the back called the optic disc. So if you look here, this is where all the axons are actually leaving. So where they are leaving, there are no receptors there. So that's basically your blind spot. Okay, that's your optic disc. If light happens to fall on that optic disc, you won't see it. Okay. There's also a whole lot of blood vessels in this area. So, obviously nourishing different parts of your eye. You would have seen this in the sheep eye. So that really beautiful iridescent blue that you saw is a structure called the tapetum lucida or tapetum lucidum. Either term is fine. And this is the reason for eye shine in animals. So here you can see this is a black-footed ferret, and you can kind of see that nice bluish kind of yellow eye shine coming from it. Now here's where we differ. Okay, so let's kind of just go over something real fast. When light enters our eyes, right, the back of our eye is dark like this part of the choroid is. Okay? So basically, light's going in the back of our eyes, it's hitting the retina on that one spot that we want it to, and then this choroid absorbs any of that extra light, right? And by doing so, it prevents any kind of scatter of the wavelengths of light, okay? And by preventing scatter, we see very nice, sharp, crisp images, Okay, so that's going to be very important to us. We like to see sharp, crisp images. That's why I wear glasses, right? Or you might have contacts or whatever. Now, for an animal that is predominantly active at night, what it cares about is night vision, right? Being able to see under very low light conditions. So these animals will have this tapetum lucidum. So basically, when light enters into its eye, it also bounces back out. So giving those photoreceptors twice the opportunity to pick up on the wavelengths. Okay, so not just coming in, but also bouncing back out. Now, unfortunately, this causes scatter, so their vision is not nearly as sharp as ours would be. However, for the most part, that's irrelevant, considering that all you care about is seeing what you're hunting, not so much, well, is that Bob or is that Janine? You can't tell. Let me squint. Is that a C or a G? They don't care. They don't need to see that clearly. They just need to know that something is there, right? Either to hunt it or to get away from it, okay, from being the prey. So that's what this tapetum lucidum does. All right. So when we process information, visual information, what we care about is refraction of light, so the way the light is being bent. Again, we're trying to focus it on one spot in the back of the eye. And that bending of the light is being done by the cornea and the lens. So again, all these light rays are going to have to fall not just on the retina, but on a specific area in the retina. We'll see that in a second. The lens has to accommodate, right? We mentioned it has to go from being kind of thicker to thinner, right? To focus light, whether or not we're close or far away. And then the pupil is also going to either constrict or dilate, depending on how bright the light is, around us. Right. So here's refraction, right? We all know this. This is light, bending of light. So if it goes from one media to another, it changes the speed, so it bends. Okay. So in the eye, light is refracted at the anterior and posterior surface of the cornea, as well as the lens. All right. So here's an image. So, and hopefully you can see this. So here's a tree, you're looking at it far away. So this is distant viewing, right? Parallel rays are coming in, so this is what you're seeing. Light waves are coming in, being bent by the cornea and the lens, right, onto that back part of your retina. Okay. Here you're looking at something up close, and here, so there's like a little thumbtack you're looking up close. And if you'll notice that lens is going to be much thicker. So again, it can bend those light rays to hit that exact same spot in the back of the eye. Right? Interestingly, the image is actually inverted, so left is right and right is left, and upside down. Okay, so your brain, however, knows to take that information and flip it. They actually have glasses where it flips the image back, and when people wear that, I forget how long it takes exactly. They wear that for a while, and they see everything upside down and backwards, right? And they'll keep seeing it until it's almost like a switch flips, and all of a sudden, poof, everything is right side up, and left is right and right is left. I'm sorry, left is left and right is right. So everything is normal. And if they were to take off their glasses, everything would look upside down and backwards again, and the brain has to readjust to that. Isn't that wild what your brain can do, right? So again, to get these crazy glasses, brain will adjust to that. Take them off, brain will adjust to that. It's amazing. Okay. Most of that refraction, so that bending of the light, is going to be done by the cornea, and the fine-tuning will be done by the lens. Okay. You may have heard of these terms. So myopia is being nearsighted, meaning you can see clear up close, but you cannot see clear far away. In fact, you may end up hearing this term at some point, in your life. If you ever hear someone who's myopic, that's an insult. What you're saying is that person is very nearsighted. They cannot see the consequences of their actions far away. Right? Hyperopia is being farsighted, meaning that you can see things far away very clear. However, up close you might need reading glasses. And then an astigmatism is some sort of abnormality, either due to the curvature of the cornea or the lens. So they're not perfectly smooth. They may have divots or kind of like waves in them. So that'll affect how obviously light is being bent. Okay. So here you can see this is what we do to correct for vision. Okay, so here's someone who's nearsighted, right? And what ends up happening is that those light rays, like the eye, basically the shape of the eye is wrong. So the eye is too elongated. So the cornea and the lens are trying to focus the light here, which is where it should be. However, the eye is actually way back here, so it's not focusing it on the right spot. So you get the right kind of lens, that corrects the vision, so everything is nice and crisp and on that part of the retina that you want it to be. When you're farsighted, it's exactly the opposite. The shape of the eyeball itself is a little bit too short. So again, it's trying to, the lens and the cornea are trying to focus things on the back of the eye where it should be, but it's, too close, so the light rays are still scattered. So you put a lens in front of that and adjust it so that it hits the back of the eye where it should be. All right. Hopefully all of that made sense. We're going to talk a little bit more after this about exactly how that retina functions and how we see. Okay, so I'll see you next time.